Here's our first Corona Apocalypse lecture, Chapter 4. This is all about genetic variation, as you can see here. And genetics is all about genetic variation. So changes in DNA sequence that lead to changes in phenotypes. That is the basic of all biological variety, why we're all different from each other, why we are different from other organisms, all that kind of stuff. This chapter on the schedule, we've got 3 lectures to cover this schedule, and we will I don't know if it'll take 3 full lectures, but I'm not going to do it all in one presentation. I'm going to break it up and to try to keep it to our normal class time, which is about an hour. So that should be more than enough time. There's a lot of info in this chapter. It's not the so we'll try to break it down to what's important. The DNA damage and repair stuff is always a little complicated, but I think the repair is more important than the actual damage stuff. But we'll take a look at it all. So what we're looking at on this is how DNA damage occurs. We'll look at a couple of different types of damage that can happen, how DNA is repaired, and the idea is that DNA damage happens all the time. The sun damages your DNA. Just DNA replication damages your DNA, and the DNA has to be repaired, and it's repaired a lot. It's like 99% of the time, 99.9% of the time it's repaired. But sometimes it doesn't get repaired, and then those DNA changes become permanent, and that's when you have a mutation. But if you have a mutation, it has to be, in order for it to sort of persist in a species, it has to be selected for, or at least not selected against. And so mutations and their appearance and their frequency in populations is all related to evolution. And so there's going to be mutations that are going to be either selected against, which is negative selection, or selected for. If they provide some sort of advantage, there's positive selection. So we'll look at that a little bit. We will look at normal variation. Normal variation is usually called polymorphism. We've seen that term a little bit, and we'll see it again now, as opposed to some kind of mutation that leads to disease or something like that. And Chapter 7 really goes into genetic diseases, and so we'll leave most of the diseases that are caused by mutations till Chapter 7. We will talk about diseases that are due to the inability to repair DNA. There's quite a few diseases, including cancer and other diseases, that the main problem is that you can't repair DNA, so you accumulate a lot of mutations, and that's bad. Alright, so how variation is measured. I don't know that we're going to look at that too much. One thing we'll look at is something called a SNP array. It's a SNP, like a microarray, and so we're actually going to do a SNP microarray on CASEIT later. So we'll take a look at one example of those in this presentation. It's all about genetic variation. So here's some type of damage that can occur. You can actually break the DNA. So over here we've got a double-stranded helix over here. You can break that DNA. You can do a single-stranded break. You can see right here this little black strand is broken, or you can have what's called a double-stranded break. So we'll call this a single-stranded break and a double-stranded break, and we will look at those in some detail. You can so this example is a example of so B over here is an example of a base deletion. It's a little bit hard to see what's going on here. But this base, so this is the base, and that's missing here. So we're losing it. It's going away, and we're losing the base. Notice that it's just the base, so it's not the whole nucleotide. A whole nucleotide is a base, and a sugar, and a phosphate. So that's a nucleotide. And this is a base. So that's something we'll want to keep in mind. Base modification over here. You can see you can modify bases, chemically modify them, oxidize them, deaminate them, things like that. You can just chemically modify the base. And then here's an addition of a big, huge thing to the DNA. So all of these things can damage DNA. This one is particularly common. That's thymine. So this is called a thymine dimer, and that's caused by UV light. So sunlight causes thymine dimers, which is a damage in DNA, and there's specific DNA repair mechanisms for that. And sometimes you can cross. This is two examples of so a dimer means that two thymines are linked together here, so they're cross-linked, and then you can cross-link other things. There's another example showing cross-linking. So these are various types of damage. We'll take a little closer look at them. And so they're summarized here. You got DNA strand breakage, single-stranded breaks, double-stranded breaks, base deletion, which is just the base, not the whole nucleotide. I don't know, it's always easy to see these typos. So nucleotide. And the sugar phosphate backbone is still there, and but the base is removed. Base modification, there's tons of different ways to modify a base, and then you can cross-link bases. We're not going to look at all the different ways to modify them. It's just, if you can remember the main category, that's okay. This is just a closer look at strand breakage. This is really common. This is an important type of damage, and particularly the way these things are repaired is really important. So we'll spend a little bit of time looking at this. Another way to maybe draw this would be, if you've got double-stranded DNA, you can break one strand, or if you've got double-stranded DNA here, you could break both strands. So we just kind of break it in half, and then that has to get repaired. Pretty obvious. Single strand break, double strand break. Base deletion, this is just a close-up of that. So this big base, whatever it is, it's probably guanine. I don't know. I know it's purine because it has two rings, and it gets removed. Oh yeah, right, look at that. It's a guanine right there. So now what you have is you still have your DNA backbone, sugar phosphate backbone, but you're missing the base. So that means it's an abasic site, and it's usually purines that are missing, and so that's called apurinic site. A means without, so now we're missing, and so we're missing a base, or we're missing a purine. So that's a particular type of DNA damage, and there are specific DNA mechanisms, repair mechanisms, that can see those types of damage and fix them. Base modification, just lots of different ways to modify them. This in particular is formed by oxidation, and we're not going to get into the chemistry of it really, but just know that a lot of things cause you've heard of like free radicals, and a lot of things cause oxygen free radicals and that's why antioxidants are good, because oxidants are bad. So if you have oxidants in your cells, or generate them in your tissues, those oxidants will oxidize DNA and will cause DNA damage. So that's why people think antioxidants are good. Things that really are known to cause mutations are things like X-rays, smoking, and the reason these things cause so much damage is they generate oxygen free radicals, and what they tend to do is they can make these oxidative changes, but they can also break DNA. So these oxygen free radicals can all right. So my microphone stopped working on the last one. Hopefully it'll keep working on this one. At least it gave me a little warning as to that the microphone wasn't working, so I didn't keep talking the whole time. So basically we just left off with that oxygen is bad and it can break DNA. That was back on this slide. And now we'll move on to this one. And this is just saying UV light can cause thymine dimers or thymidine dimers. So if you looked at that in DNA, you had two T's next to each other. They form a covalent bond here. So this is in the context of DNA. And so then the DNA is not going to pair up very well. Even if there's an A over here, it's going to look weird. And then when the cell tries to replicate that DNA, it can lead to a mutation. Alright, so there's too much here. This is just all kinds of, this is everything all summarized into one slide of all these different types of attacks that can happen to DNA, and we're not going to worry about that too much. We don't need to get into all the details of every kind of DNA damage. What I think is more interesting is DNA repair, because you have to be able to repair the DNA or everything is bad. There's a lot of diseases. It can lead to cancer. It can lead to cell death. So you have to be able to repair it. All right, so we know about, these are all the main types of DNA repair mechanisms. The one hopefully you've heard of a little bit is DNA proofreading. We'll take a look at that. That just happens during, not doing, during DNA replication. So there's a little DNA damage right there that I failed to repair, and therefore that's a mutation because it persisted into my PowerPoint slide. But we'll talk about DNA proofreading. Base excision repair. So we looked at what happens that you can actually remove a base. So we'll take a look at that. Nucleotide excision repair, where an entire nucleotide is removed. Mismatch repair, which is kind of an interesting one. And then of course when we break the DNA, we have to repair it. And there's two types of DNA breaks: single-stranded and double-stranded. And we're going to focus on the double-stranded DNA breaks. So this is proofreading, and this happens during DNA replication, and it's relatively simple. So you know that in DNA replication you need a template, you need a primer, you need a polymerase, and you need some DNTPs. See, so this is why I made you remember that, because it comes up all the time. Whenever you're talking about DNA replication, PCR, DNA repair, you need to know the basics of DNA replication. So this is what happens. So we got our template, got our primer, and the polymerase is cruising along making new DNA. And then let's say there's, in this case, they're showing a C and an A that is messed up, and then it continues along. And then here's the polymerase, and it looks back here. It says, wait a minute, that's no good. Why did I do that? I screwed up. And so then, let me make sure that, so this 3' goes there. And so it actually can recognize that it has made a mistake. So then it erases that. So let's say that it's gone this far, and look at my little eraser. So the DNA polymerase will just erase what it just put in. It's not going to erase the whole thing. I was afraid that was going to happen. But it erases a little bit. So here's your template. It made a mistake, and then it erases that mistake. It kind of goes backwards and erases, and then it fills it in with new stuff. So this is your template, and then it erased its mistake and replaced it. So proofreading is erasing a mistake, erasing and replacing. And so that's just like if you're typing along and you realize you make a mistake and you hit backspace. So it's a lot like backspacing, and then you retype it. So that's all proofreading is, but that happens during DNA replication, and it's a function of DNA polymerase itself. It can replace its own mistakes. So right here it erased that and got rid of that, and here it put in the normal base. So that's what all proofreading is, backspacing and retyping. Let's go to the next slide, please. There we go. Every now and then I put a slide in like this to remind myself to draw something. I already drew it, but I'll draw it again. So, template, primer, making new DNA, make a mistake. That mistake doesn't pair up right, so it forms a little mismatch, sort of. And so if we had, I think that was a C, and then it tried to put an A in there, and that's not a good match. And so the polymerase can recognize that that's bad. Then it erases some of this, so it's going to go back and erase this. It still has a template, and then it just goes back in, and here it had put in an A, and then next time it gets there it puts in a G because that's what it's supposed to put in, and then it continues on. So it's just a matter of sort of proofreading and sort of repairing something as you go. However, sometimes that proofreading doesn't work. You get a it's not 100% efficient. Sometimes that wrong base gets put in there, and you cruise along, and that mismatch stays in there. And so this is what a mismatch, you can represent a mismatch like that. That could be that A-C mismatch or something like that. Now in bacteria, so this is a difficult problem for the cell to deal with. So let's say this is our mismatch, and then the cell realizes it later. It didn't realize it right when it did it, but after replication, it goes back and says, oh, that doesn't look right. And so it has to remove the wrong base and put in the right base. This is the original, and so that's correct, and then this is the wrong one that was put in. But as far as the cell can tell, that's a normal A and that's a normal C, and so it doesn't know which one's wrong. It doesn't know which one to take out. But there actually is a system in bacteria. So here this got the 3' to 5' on the top. So this is the original template. And sometime after DNA replication, the DNA gets methylated. So this is a methyl. And so that's an H, methyl. And so when the cell sort of looks back at the DNA, it can tell that the old DNA strand is methylated and the new strand hasn't been methylated yet. New strand. And so it actually can tell by looking at the methylation status whether or not, so which strand is old and which is new. And so if there's a mistake, it's going to sort of, I'm doing a lot of what they call anthropomorphizing, saying the cell's thinking all this stuff, but just biochemically it can look at this DNA and notice that there's a damage, distinguish the old strand from the new strand, and then realize that the mistake happened in the new strand, and so it's going to get rid of the new strand and replace it. And what it does is it actually makes a little break in the DNA there, and then it removes a stretch of DNA, and then it just resynthesizes that DNA and fills it in with the normal stuff, and then sometime later both strands will get methylated. So if there was a mutation that happened like way later, it wouldn't be able to tell the difference. So these this process is carried out by these genes called MUT-H, MUT-L, and MUT-S. And so these are genes that make proteins that are necessary for repairing the DNA. And the reason I point that out is that in humans, there are human homologs of these genes, or human versions of these genes, that if you mutate those, they lead to cancer. So we'll look at those in a minute. Now, one thing that's weird is that humans don't actually use this same mechanism. Humans don't have this methyl recognition system, and so by some other, so down here it says that eukaryotic cells rely on some different method to distinguish the strands at a mismatch, and that hasn't been figured out yet. So they're trying to figure that out. Who knows? So anyway, so that's mismatch repair, and the genes that are involved are these MUT genes. They're called MUT genes because you know how if you have a mutation in a gene, then that gene gets named by what its phenotype is. So if you have a mutation in a white gene, in Drosophila, normally have red eyes. You got a mutant that has white eyes, that's called the white mutation because the phenotype is white. Here, if we mutate any of these genes, the phenotype is going to be we're going to have too many mutations because it can't repair the DNA. So MUT stands for mutator because there's a mutator phenotype. That if you don't have the DNA repair genes, you get a high rate of mutations, and so that's how those got their name. All right. So we'll talk about those again. Well, let's go back. So, and then in humans, there's MUT H homolog, MUT L homolog, and MUT S homolog. So homolog means that it's homologous, or it's the same gene in a different species. So it's sort of like ortholog. We looked at orthologs before. Homolog's a more general term for that. And so we'll see that if we have mutations in any of these genes in humans, then that leads to a mutator phenotype, and in the case of humans, that can lead to cancer. So we'll come back to that later. Base excision repair. So what we just saw is proofreading, and then we saw mismatch repair, and then this is base excision repair. So here's a case where there's a GU mismatch. That's weird, and cells can say, oh, U is not even supposed to be in DNA. And so there's a mechanism that removes that U by breaking this bond here. And then the DNA backbone is still intact, but we're missing a base, and so that's an abasic site. And then there's a particular mechanism, no matter how you get an abasic site, there's a repair mechanism that goes in and cuts out some of that DNA, fills it back in, and replaces it with DNA ligase. So a lot of DNA repair mechanisms are like that. Whoa, I just did something. Well, that's interesting. There's some crossing over. We'll come back to that later. Oh boy. I pushed a button on my super pen and all hell broke loose. So we will edit that out, but first let's see. All right, so we're back to here. All right, so abasic site, and you repair it. So here we had some kind of weird damage. We got rid of the base, so we have an abasic site, and then what happens is you cut on either side of that, and then you just fill it back in with new DNA and fix it, and then put it back together with DNA ligase. So that's base excision repair. That it recognizes an abasic site, or it actually, base excision means it removes a base. So here's a base, remove it, and then fix it. All right, so that's what base excision repair means. Nucleotide excision repair is where you remove an entire nucleotide instead of removing a base. So this happens a lot. One good example are these thymine dimers, where UV light causes two thymines to be stuck together. And then now, instead of removing just the base, you just remove the whole region. You remove a bunch of nucleotides. So it's kind of like base excision repair, but it just doesn't have that extra step of removing the base first. And then you just get rid of that DNA and then fill it in. So that's very common type of mechanism. You got some kind of mistake. It gets recognized. You cut out what was there, and then you fill it back in with the DNA polymerase, and then you tie it up with ligase. So that's just sort of a very common DNA repair mechanism. And there are diseases that are associated with pretty much all of these DNA repair mechanisms. If you can't do them, there's a lot of diseases. Now here's one that's particularly interesting. So we're going to look at homologous recombination. And so this is a way to repair double-stranded breaks. The mechanism itself is a bit hard to wrap your head around, so we're not going to worry about all the details, but you have your DNA, then it gets broken, and then that we're going to repair that. There's going to be two mechanisms for repairing that. The first one is homologous recombination. And this is a long sort of description of it, but the idea is, so we have our double-stranded break, and what's basically going on here is that information in the, here we have sister chromatids, and so that gets broken, and what we're going to do is take the information that's in the sister chromatid, which is actually identical to the top double-stranded thing, top sister chromatid, and we're going to take the information that's in the sister chromatid and copy it into that site, and that way it goes back to normal. But how it actually occurs is a little bit complicated. First of all, these strands get sort of cut back, and then this strand comes down here. You can see this strand coming down here, and it lines up with that other strand and then copies it, and then this strand comes down here and gets copied there. And so it's a somewhat complicated thing. It's very similar to what happens during crossing over in DNA recombination, but we're gonna keep it even simpler than that. So what we're gonna do is say we've got our original DNA, all right, what was that? We've got original DNA. We're going to get a different pen color. We've got a sister chromatid. Then we get a double-stranded break. Got our sister chromatid, which apparently is a slightly different color, blue this time. And then essentially what happens is, without worrying too much about exactly how it happens, is that this information gets copied into there. And so what you end up with is your original DNA strand, still getting the hang of this PowerPoint business. Look at that. Why does it keep doing that? So we've copied the blue stuff up into here, and then the bottom strand is still the same. So that's very handy. If you have, so it's like the sister chromatid acts as a spare copy of the other chromatid. So if you actually break a chromatid, you can use the other chromatid as a template to fix it, and that's very common. And so that's called homologous recombination, and it's used for all kinds of different things. It's actually super important when it comes to CRISPR. So while we're here, I just want to point out that in CRISPR, what happens is we break the DNA at a particular spot. That's all CRISPR is. We use a system that can guide a protein to a particular spot and cause that spot to be broken. Then you can repair that site, and one of the ways you repair that site is homologous recombination, and so that's one way to do it. And we can take advantage of that and modify it so that we can see how we're adding blue stuff in there. We can modify it so we can add other stuff in there instead of just the blue stuff. And so we'll come back to that, but that's why I wanted to make sure that I focused on double strand break repair. So this is one example of double strand break repair, and then the other way to do double strand repair double-strand break repair is this mechanism. This is called non-homologous end joining, or NHEJ, and it's relatively simple. Really, you start with double-stranded DNA. I'm going to get a better color. Oh, what color do I want to use? I don't know. Picking a color can mess up your whole thing. All right. We'll go with black. All right. So we've got a double-stranded DNA. Gets broken. So we're not really missing anything. We're just breaking it. And non-homologous end joining basically just puts those molecules back together. So it just takes those two things and puts them back together. So it just is gonna. It's basically like ligation. Just takes those things, pushes them together, and then ligates them. But it's not precise. So when it puts it back together, sometimes, we'll make these red, sometimes you add new nucleotides in there. So it's not precise. So these are just some of the proteins that are involved and some of the things that are happening. But the idea is that it's not precise. And so that's fine if you break DNA and you need to fix it real quick and you don't happen to have a template. So if you have a template and you use homologous recombination, it happens perfectly. It's exactly the way it's supposed to be. But if you do non-homologous end joining, it puts it together, and that's better than having these two broken ends of DNA floating around. But it can add extra nucleotides, so it can cause mutations. So in CRISPR, we break DNA. If we do homologous recombination, that's one path. Or if we want to introduce mutations, this homologous recombination or this non-homologous end joining actually causes mutations just sort of naturally. So that's kind of how CRISPR works, is taking advantage of these normal DNA repair mechanisms. Alright, so I keep putting these things to draw, but that's alright. So that's, we'll draw it again. DNA, double-strand break. Now what actually happens is when you break double-stranded DNA, it's not always just perfectly aligned like that. Sometimes, and what that other slide is trying to show is sometimes you got a little overhang. Sometimes you lose a little bit of DNA. It might get chewed off the ends. But then when you put it back together, you sort of need to. Sometimes it needs to use a little bit of glue. And that glue is extra nucleotides that kind of come in here and sort of fill that. And then this is a mutation. And that's basically it. So let me just summarize here. Well, the next slide will summarize this. But if we have a double-stranded break, we can do HR, or we can do NHEJ. If we do HR, we copy a template, and there's no errors usually, or fewer errors. But this is, you just ligate them together, and it's error prone, or it's not precise, so you get mutations. So just keep that in mind. When we talk about CRISPR, we'll look at some specific applications of this, of how you can either add new DNA to a spot or add mutations to a spot. And that's what this slide is showing, is that you get your double-stranded break, and you could either do the NHEJ pathway or the HR pathway. NHEJ, you've copied, you use this green template to copy the black. It doesn't really show it that well here, but you end up copying one sister chromatid's information into the other. And these are just some of the proteins involved. This is molecular biology, so you always get blobs and acronyms that show everything, but we don't need to know all the names of all those things. Then here's a nice little description of it. So that's kind of cool. And so I might, this is a thing. This is one of my things. So this is something that I'll figure out a way to quiz you on one way or another. All right, so here's a summary of all that stuff. We can so there's these different types of DNA damage. You got a base mismatch. You use mismatch repair. Sometimes if you have a small type of DNA modification, you can cut out the modified base and use base excision repair. If it's like a big thing, so this is like if there's like a smaller, just like an oxidative change or something like that, just a small change in the base, you can just get rid of the base. If it's a big thing, so like this, the intra-strand cross-link, that's like when those two T's get cross-linked together. We can use nucleotide excision repair. Single-stranded breaks, we didn't really talk about that. And then double-stranded breaks, you can use HR or NHEJ. And so that's just a summary of the different mechanisms. And then this is interesting. So this is a bunch of diseases that are associated with defects in DNA repair. And so this is the part that I think is pretty interesting. And so here, let me take a look at see if we can. Oh, I didn't put the notes in there, so I was just curious as to what I forget what these things stand for. So we can. It doesn't really matter, but we can look at that in the book. But it's not that big of a deal. So anyway, so certainly don't have to memorize all of these different diseases. This book is based on human genetics and human medicine, and so whenever possible, we like to point out the diseases that are involved. And this just shows that there's a lot of diseases that are involved. Now, one of them I've already mentioned. So mismatch repair leads to, this is a nice big long name, hereditary non-polyposis colorectal cancer. So that is hereditary non-polyposis colorectal cancer, HNPCC. In other words, colon cancer. So it's a type of colon cancer that is inherited. And the reason it is inherited is because, well, it's a little bit complicated, but the genes that are mutated in this type of thing are the MUT-S homolog and the MUT-L homolog. Remember, those were the genes that were involved in mismatch repair in bacteria, and the human versions of those genes are mutated, and that leads to this type of colorectal cancer. Xeroderma pigmentosum is XP. This is a problem with nucleotide excision repair, and so that means they can't repair UV damage. And so we'll show you some pictures of this, but this shows up in the skin a lot. So when people are exposed to sunlight, DNA damage occurs, in particular those thymine dimers, and then this nucleotide excision repair pathway can't fix them. And so that's that's called xeroderma pigmentosum. And then there's all these other problems with different DNA repair mechanisms. Let's see. This one's interesting. This is a premature aging syndrome. So we'll take a look at that, because the cells can't repair DNA damage, so they age faster. Double-strand break repair leads to something called severe combined immune deficiency, or SCID, and we'll talk about that in a little bit. That's it turns out these DNA repair pathways are important in some immunofunctions. So I never quite know what to do with this. I certainly don't expect you to memorize it all, but for all of you folks that are interested in going into medicine, read up on it and see what you think. The idea is that if you have DNA damage and you can't repair it, it leads to diseases. And the C here, I'm sure, means predisposition to cancer. So all of these things, if you have DNA damage, it's very common for them in those syndromes for you to be more prone to cancer. Another thing that's interesting is this ataxia stuff. Ataxia means uncoordinated movement. Here's another ataxia. So that's kind of weird. I've never gotten a good answer as to why the DNA repair affects this. So that ataxia means like uncoordinated movement, like loss of neuromuscular control. And this is a good one: ataxia telangiectasia. So I don't know what that stands for, but that's genes that are involved in this double-strand repair lead to that disease. So once again, don't have to memorize it, but be familiar with the concept that DNA repair. Why does DNA repair lead to disease? Why does lack of DNA repair lead to cancer and things like that? All right, so here's a couple of examples. A is xeroderma pigmentosum, and this person has a lot of these growths on her face or his face, and a lot of times those lead to melanomas, and that's basically because they don't have the ability to repair so nucleotide excision repair, so they can't repair UV induced thymine dimers. So this is UV induced, so they're very sensitive to the sun. Accelerated aging in Werner syndrome. I'm not exactly sure how B exhibits that, but there are some premature aging syndromes. And C, go ahead and you can read more about this in box 4.1. I don't really usually get into the details of these syndromes. I'm not a doctor, so I don't know that much about the actual medicine behind them. But I just want to point out that these things are. We'd have to look at this box to see what the actual ages are. And so the question is, how old is this person? So this is her when she was younger. This is her when she's older, but she looks pretty old, and we'd have to look that up. Actually, I'm going to pause right now and go look that up. Okay, so she's 13 years old here, and 56 years old here. Just for reference, I'm 57. So make of that what you will. She looks a lot older than 56, and that's the point. All right, so I'm going to stop here. I have to give up my home office workspace to my wife, who has to have some interviews with some people, and that's been about an hour. So putting the two together. So we'll just stop it there and pick it up in a bit. Also cause double-stranded breaks, and so